Alright, so in the last video I got cut off a little because my recording software only records up to 10 minutes. So, hopefully this one uploads right after the one before it. So, what I was talking about is how these centrosomes generate these spindle fibers, and those spindle fibers make the centrosomes go to opposite ends of the nucleus that's currently breaking down. So, they're not completely here yet, so they're on their way though. And personally, I find prophase the hardest to remember, and that's because besides interphase, oh, hold on, let me make sure. Okay, besides interphase, in terms of the phases of mitosis, cells spend most of their time in prophase, and that's because all this stuff happens in prophase. And all this stuff, once again, just to conclude it, is chromatin condenses into chromosomes. These centromeres start pushing against each other to go to opposite ends, and the nuclear envelope starts to break down. Okay. I didn't finish writing that, but that's okay. I think you guys get the drift. Alright, so the next phase of mitosis is metaphase. And in metaphase will always involve the lining up of chromosomes, even when we talk about meiosis. So here we go. Here's our cell, and I'm not going to draw a nucleus this time because by now that nuclear envelope that we saw in interphase that was breaking down by prophase is completely gone in metaphase. So all that happens in metaphase is that the chromosomes line up. Maybe we have a smaller one from our dad. Or sorry, red we said was for mom. We shouldn't forget the ever so essential, that's too big, the ever so essential centromeres, which are in green. Centromeres basically just connect these two chromatids. So they line up on this plane, and that's all that happens in metaphase, really. And then the next phase is anaphase. Oh, actually, forgot to mention something here. These centromeres that we saw that were replicated in the S phase of interphase and started to generate spindle fibers in prophase have succeeded in pushing themselves to opposite ends of the cell. I'll also draw their spindle fibers. So the pink are the centrioles inside the chromosomes. Sorry, not the chromosomes, the centrosomes. Hope you guys don't get that mixed up like I just did. And these spindle fibers are being pushed out from the centromeres, from the centrosomes towards the centromeres. And we'll see why that happens right here in anaphase. So anaphase, scroll up a little. Whoops. I don't know what it is with my little keyboard, but it keeps acting up. I really need to get that fixed. Anaphase. So in anaphase, we have our cell again. Let's make it a little larger so we can see what's going on. And in the cell, by now, these centrosomes that we've been talking about for so long have succeeded in pushing out their spindle fibers. Ooh, that's the wrong thickness. Have succeeded in pushing out their spindle fibers and attaching them to the centromeres that connect the chromatids. And some of these microtubules also connect these two centromeres right here. Sorry, these centrosomes. I keep mixing that up. And what that does is that these microtubules push against each other and make the centrosomes go in opposite directions. And we'll see why that's important in the next phase, which we'll get to in just a second. So here's one chromosome, here's the smaller one, and then here's our homologous chromosome from our dad. There's our smaller homologous chromosome, and the ever so important centromeres. Then we can continue drawing the spindle fibers. So they latch on to this part in the chromosome known as the kinetochore, in the centromere known as the kinetochore. And they push, these microtubules are going to push apart these chromatids so that in telophase, what's happening, so that they go to opposite ends of the cell, which happens in the next phase, which is telophase. How much time do I have? Okay, I have 4 minutes left. That's plenty of time. So in telophase, what ends up happening is that what had previously been, this was yellow, what had previously been our one cell with our, what had previously been our one cell, let me redo that. With our centrosomes on each side, has now turned into 2 cells. Well, actually it's not quite 2 cells yet, but it's almost to the point where you can consider it to be 2 cells. Let me use white. I haven't used white at all. Alright. So this one big cell is beginning to turn into these 2 smaller cells. I'm gonna have to make some more room right here. 2 smaller cells. Just give me a second to draw this. And what happens here is a process known as cytokinesis. And cytokinesis is basically when the cytoplasm of the cell is pushed in from either direction, right here. I'll do it in red. So the cytoplasm is pushed in, and you can see how this cytokinesis itself is not technically telophase. It occurs right after telophase. But what does happen in telophase is that these chromosomes that we had up here before, let's just scroll back to anaphase. In anaphase, where we had these chromatids joined from the centromere, the chromatids are finally pushed apart and are at opposite ends of the cell. So we'll have that big one from our dad. That's too thick. That big one from our dad, and then that, from our mom, and then that little one from our mom. Then we'll have that big one from our dad, and then that little one from our dad. And then we'll also, on the other side, we'll have the same thing, because remember, they got pushed to opposite ends of the cell. Whoops, wrong color. So have another big one from my mom. And then we'll have a little one from the mom. Big one. A little one from the dad, sorry. That should be orange, not yellow. So basically in telophase, the centrosomes also go to the... Well, oh, right. The centrosomes end up over here. I almost forgot to mention this. But the nuclear envelope starts to reappear. So let me just show that really quickly. I think we did it in blue up there, so I'll continue. The nuclear envelope's being built up again. So the nuclear envelope's sort of a temporary thing. And these centrosomes with their centromeres in the center are on opposite sides. So that when these cells finally do split, right along here, each cell will have each nucleus will have a centrosome to accompany it.